Hi, good morning. I'm Aditya Dutta. I'm an assistant professor at the Department of Animal and Food Sciences with a joint appointment at Biological Sciences at the University of Delaware. And today I'll be talking about targeting mitochondrial metabolism and using it as a tool for precision prevention in prostate cancer. So, prostate cancer is extremely common. Um, when we look at estimated new cases, prostate cancer is the number one cause of cancer amongst males in the United States, and it's the second largest cause of death due to cancer amongst males in the United States. Therefore, any sort of change in the disease landscape has far-reaching effects for a large percentage of the population. A really interesting thing about prostate cancer is that it has a really long latency. What I mean by that is that a lot of individuals, after they are first diagnosed with the prostate cancer, live with prostate cancer. A majority of them do not die because of prostate cancer. Moreover, more than 50% of individuals who are diagnosed with prostate cancer do not need treatment for prostate cancer. However, more than 90% of individuals who are diagnosed with prostate cancer are treated for prostate cancer. As you can imagine, that is clearly a problem because there is overtreatment. So prostate cancer, similar to most of the cancers, it starts off as a normal epithelial structure. So the prostate is part of the male reproductive system, and it produces semen, which helps the sperm to thrive. So it has a lot of ductal physiology. And what you see over here are these epithelial cells with these basal cells on the side, and there are also some neuroendocrine cells. Over time, you get what we call as pre-neoplasia. That means that some of these epithelial cells start to divide. This pre-neoplasia is seen to be the precursor for prostate cancer. This then leads to adenocarcinoma, that then leads to clinical manifestations and can be clinically identified, which leads to full-blown metastasis. Over time, a lot of these processes and a lot of these genes have been identified which are involved in prostate cancer. Some of these processes involve inflammation, oxidative and DNA damage, senescence, and most importantly, the development of castration-resistant disease. What I mean by that is that the prostate is controlled in part by hormones, especially androgens. The idea is that more androgen leads to greater levels of prostate growth. However, after a certain point in time, when the disease moves to castration-resistant disease, even reduction in the levels of androgen does not control the spread of the disease. Some of the genes that are involved in prostate cancer are NKX 3.1 downregulation, MYC overexpression, PTEN inactivation, MAPK activation, as well as EZ2 overexpression. My talk today will be focused on NKX 3.1 downregulation. And when we talk about NKX 3.1 downregulation, what we're really talking about is NKX 3.1 loss of function. So NKX 3.1, as you can imagine, given the fact that it's labeled right here, is involved with the initiation of prostate cancer. It's one of the first genes to be functionally lost, and it's involved with inflammation and oxidative and DNA damage. NKX 3.1 is a prostate-specific gene. There are other NKXs in other organs, like NKX in 2.1 in the lung, 2.5 in the heart, but NKX 3.1 is specific for the prostate. So about 5 years back, in 2016, we identified that NKX 3.1 is absolutely crucial for formation of the prostate. Not only does it allow formation of a full grown prostate, you are able to take tissue, a fully adult tissue from a different dermal layer, introduce NKX 3.1 into those cells, and under certain conditions, you can engineer a fully grown prostate. This process requires G9A as well as UTY, and NKX and G9A come together to control UTY expression, which leads to the development of the normal prostate. So effectively, you can now design a fully grown prostate in the laboratory. Given the critical role of NKX 3.1 in this process, we reason that NKX 3.1 should be able to identify and have some sort of disease relevance. So to look at that, we looked at humans that had high levels of NKX and humans that had low levels of NKX, and we compared them to mouse models with high and low levels of NKX 3.1. We looked at the expression profiles and we performed what we call as gene set enrichment analysis, which allowed us to identify an NKX 3.1 dependent signature. And using that signature, we were able to classify groups that would respond to particular drugs and would be beneficial for those prostate cancer patients. Now, once we knew that we could use NKX 3.1 to classify patients, what we did was that we wanted to see what else can it do. We next identified the relation between NKX 3.1 and inflammation, and then we started to look at NKX 3.1, the protein. So over here, you have NKX 3.1, and NKX 3.1 has this N-terminal domain, it has this homeodomain, and it has a C-terminal domain. NKX 3.1 is a nuclear transcription factor. That means that it functions in the nucleus and it binds DNA. So to bind DNA, it uses this homeodomain. We were also able to identify multiple polymorphisms of NKX 3.1. We do not call them mutations, we call them polymorphisms because they're part of the general population. One of them is the R52C polymorphism, and the other one is the T164A polymorphism. These are present up to 14% in the general population. Now, what's interesting about these polymorphisms is that individuals who have the T164A polymorphism tend to have a higher incidence of prostate cancer. Individuals who have the R52C polymorphism tend to not have a higher incidence of prostate cancer, but once they do get prostate cancer, have a more aggressive form of the disease. Now that is absolutely critical because there are certain populations, especially the African American population, which not only is at a higher risk of developing prostate cancer, but when they do, their disease is much more aggressive. So we asked whether there was we could identify why and how this R52C mutant and this T164A mutant could be leading to more aggressive disease. So what we found was that NKX 3.1, as I mentioned, was a nuclear transcription factor. However, last year we published and we showed that under conditions of stress, this nuclear transcription factor actually moves into the mitochondria. When it does move into the mitochondria, it actually binds the mitochondrial genome and controls mitochondrial gene expression. This T164A mutant is able to move into the mitochondria, but is not able to control gene expression. Because it's not able to control gene expression, you have that higher incidence of prostate cancer. Now, what's interesting is that this R52C mutant, under conditions of oxidative stress, loses its three-dimensional structure, because of which it cannot enter the mitochondria. Because it cannot enter the mitochondria, it cannot bring about the effects that NKX 3.1 has in the mitochondria, and thus you develop far more aggressive disease. So once we knew this, we asked, can we stratify patients who have low NKX and low levels of mitochondrial gene expression? And what we see is this amazing, Kaplan-Meier. We were extremely surprised when we saw this, which leads to this huge differential when you have low NKX and low levels of mitochondrial expression. This indicates this particular Kaplan-Meier is with relation to biochemical recurrence. We've also seen the same effect for overall survival of patients. So this then takes us to our further studies. So now we've changed our question. Earlier our question was, does NKX 3.1 function outside the nucleus? Because we would see staining of NKX outside the nucleus. Now we know that NKX does indeed move outside the nucleus. So now that we know that NKX 3.1, a nuclear transcription factor, has a role in the mitochondria, what does it do in the mitochondria? Our hypothesis is that it is probably multifactorial, and it does multiple and plays multiple roles in the mitochondria. What we're asking is that, does NKX 3.1 affect mitophagy? We have seen and we have preliminary evidence to show that loss of NKX 3.1 leads to mitophagy. And does NKX 3.1 mediated mitophagy enable aggressive disease? This is aim 1 of my Delaware in-Bray pilot. Aim 2 asks whether NKX 3.1 affects mitochondrial dynamics. Does it lead to fission and fusion of the mitochondria? We do have preliminary evidence to show that loss of NKX 3.1 leads to a breaking up of the mitochondria and leads to a lot of mitochondrial fission. And if so, does NKX 3.1 mediated mitochondrial dynamics enable more aggressive disease? Now, the reason we're interested in this is that we want to go back to that earlier population that we talked about. We have all of these patients who are developing prostate cancer, but not all of them develop aggressive disease. What we want to identify are markers that will allow us to specifically identify patients, similar to what we have done till now, patients who will develop aggressive disease, and we can identify them using biomarkers much before they show clinical disease. So the tools that we have on hand are the cell lines. We use LinCap cells, RWP1 cells, and 22RB1 cells. These are both prostate cancer cells as well as normal cell lines. We have mouse models where we have loss of function of NKX 3.1, we have loss of function of P10, we have activation of KRAS, and we have deletion of P53. And we can mix and match these. We have constitutive mouse models and we have inducible mouse models. We have access to human tissue samples. We have access to tissue microarrays as well as FP samples, both with our collaboration with Columbia as well as with Weill Collins Medicine. And lastly, we have developed this human organotopic culture system where we harvest fresh radical prostatectomy samples from the surgery suite. We are able to collect them and then we can grow them in the lab for an extended period of time, which allows us to practically work on pathologically defined prostate cancer samples. So using all of these tools, we hope that we will be able to answer whether NKX 3.1 mitophagy and mitochondrial dynamics can provide tools for precision prevention of prostate cancer. And with that, I'll be happy to take any questions.